Bismillahirrahmanirrahim. So the topic of today's discussion is gram-positive bacteria, because so far we have discussed two volumes uh, of gram-negative bacteria. Rather, um, we discussed volume 2 and not volume 1 as yet. Right, so far we have completed volume 2, and now we are moving towards volume 3 and 4, which are basically the volumes of gram-positive organisms. So these volume 3 and volume 4 of Bergey's manuals of systematic bacteriology so they are basically in the second edition of Bergey's manual. So according to that second edition, volume 3 and 4 can be divided into two different groups, the Firmicutes and the actinobacteria. So these gram-positive organisms, they are further grouped into two major groups, and on the basis of their G+C ratios. So basically the Firmicutes are those which have low G+C ratios, and the phylum Actinobacteria comprises of the bacteria which have a high G+C ratios. So we'll discuss Firmicutes at a first glance. I must say that volume 3 is basically based on the phylum, the Firmicutes. Right, so first we'll discuss volume 3 and then we'll discuss volume 4, or the actinobacteria. So if we discuss volume 3 or the Firmicutes together, those organisms which have low G+C content along with their gram-positive reaction, so they are called as Firmicutes, and there are different phylums that we will discuss in this group. We will discuss some important phylums, and you will be surprised to know that most of your gram-positive organisms that you have heard in your previous classes, they belong to this group of microorganisms, that is Firmicutes. Such as you know about Clostridium, you know about Bacillus, you know about Streptococcus, you know about Staphylococcus, you know about Enterococcus, you must have heard about Lactobacillus, Mycoplasmas. These are all the part of the Firmicutes. So different orders we will discuss. So first order that we are going to discuss in this phylum is the Clostridiales, as its suffix indicate that it is representing the order. So order Clostridiales. So in this order, this order is particularly important for its endospore formation. Basically, there are two major groups that are capable of forming endospores, and one of that group is belongs to this order Clostridiales, and the other group belongs to the order Bacillus, which is also the part of the Firmicutes. So we are first discussing the order Clostridiales. So one of the most prominent genus is Clostridium, and they are differentiated with that of endospore forming bacillus because of their ability to grow without the requirement of oxygen. So they are obligate anaerobes, and they are again by shape they are the rod shaped cells, and they have endospores which is usually found at one end of the cell. So it is present at the one end, and the end becomes swollen. So this type of endospore exists in the Clostridium. Clostridium is well known for its ability to cause different infections like tetanus, which is caused by one species which is known as called Clostridium tetani, and then the famous type of food poisoning, or you can say botulism, which is caused by Clostridium botulinum. So another member is responsible for gas gangrene, which is caused by Clostridium perfringens and the other bacteria. So there are some important species that we have discussed, and another important species is Clostridium difficile, and it causes serious diarrhea, but this diarrhea is associated with antibiotic therapy because it happens only when uh, the antibiotics they alter the normal intestinal microbiota, which are also providing some benefits to your gut, and they compete for the pathogens. So whenever the antibiotic therapy it alters this normal inhabitants, these normal inhabitants, what happens that it Clostridium difficile, it it predominantly grows there, and then it produces toxin, and that toxin is responsible for that diarrhea, which is also known as antibiotic associated diarrhea, which is caused by Clostridium difficile. So basically this is an opportunistic pathogen which is a normal inhabitant of intestinal tract, but once it gets opportunity in case of the antibiotic therapies, so and only then it can produce toxin and then causes serious diarrhea. Then we have Apolopicium, and we have discussed it this organism when we were discussing about the size. As you can see that Apolopicium, it belongs to this group. It is gram positive organism, but as you can see there is a size comparison of Paramecium with that of Apolopicium. So Paramecium is far smaller than Apolopicium. So it is one of the giant, you can say, in the bacterial cell. So its shape is cigar shaped, and it was first found in the gut of red surgeonfish, which is found in the Red Sea, and it maintains a symbiotic relationship with the fish, gut of the fish. And previously, it was considered as a protozoan because of its size, but now 16S ribosomal RNA studies they placed this organism into the bacterial branch, and it can be seen with unaided eye because its size is over half a millimeter in length. So this could be easily visible by unaided eye, and it resembles the gram-positive bacteria of genus Clostridium. So basically, the order that we have started is Clostridiales, and one genus we discussed Clostridium, and Eupolopicium is another genus that we are discussing under this Clostridiales order, right? And as it suggests that it has close relationship with genus Clostridium, and definitely it has some differential remarkable feature of uh, their size. So it should be discussed here, but it has some other properties which are very different from the bacterial domain, and one such is that they do not reproduce by binary fission as which is a common mode of reproduction in bacteria. They do not reproduce by binary fission, and we have discussed it in the previous classes as well. And secondly, it has much large or huge amount of DNA, and 25 times as much DNA as human cell, and as many as 85,000 copies of at least one gene. So this is very unique on this, unique about this organism. So then we have Bacillus, the order, which is very common around you, or which is the members of which they you encounter them frequently in your daily lives. So the second order that we are going to discuss is Bacillus, and one of the prominent genus in this order is Bacillus, and it is again they are the rods. They produce endospores. They are basically non-pathogenic, but very few are pathogenic. So we'll discuss those pathogenic strains. So they are also producers of many antibiotics. They produce so many antibiotics and which are commercialized and are being used. And one of the pathogenic strain is Bacillus anthracis, and it causes anthrax. And anthrax is a disease of cattle, sheep, and horses, and definitely it's a zoonotic infection, means it can be transmitted to humans. So Bacillus anthracis and the other types of Bacillus, they usually produce endospores which are centrally located. So this is one of the remarkable uh, differential property, that you can differentiate the rods of Clostridium with that of Bacillus by looking at the place or the position of the spore within the cell. Then by Bacillus anthracis, definitely it can target the animals as well as the human, and its pathogenicity is much higher. So it can be an agent of biological warfare, biological weapon. As weapon, it can be used, and as you can see, uh, that there are some other strains as well, and one of that strain is Bacillus thuringiensis. So this strain is also important in that sense that Bacillus thuringiensis, it produces toxins which are also known as Bacillus thuringiensis toxins or Bt toxins, and it is insect pathogen. It's the best known microbial insect pathogen. So it produces toxins along with the endospores, and these toxins basically, and they target the nematodes or the insects which affect the plant crops, or particularly cotton. So what happens basically, this toxin, it perforates, or it perforates the intestine, insect intestine, so resulting in the killing of the insect. So now this organism has been commercially used. And how it's used, its toxin is basically purified from this organism, and this toxin is basically sprayed on the plants. And through genetic engineering, a crop of cotton was engineered in which the gene of this toxin was expressed in plants, and the plants started expressing that toxin, which is toxic for insects, and in this way they could save or protect themselves from the insects attack. So basically they are sold in the gardening supply shops to be sprayed on plants. So this is a well-known microbial insect pathogen. Similarly, Bacillus cereus, it's also well known for its food poisoning, and especially the starchy foods like rice. So these are the important strains. The rest are mostly non-pathogenic. So these strains, they should be discussed because of their remarkable pathogenic status. And then we have Staphylococcus. Staphylococcus is an important genus of the order Bacillus, because right now we are discussing the order Bacillus and we are discussing different genus. So in the Bacillus we discussed first Bacillus, and then we are discussing Staphylococcus. So I hope that it would be surprising for you, because the name Bacillus, or whenever we say Bacillus, so it represents just something in the dot shape. But this is the case that this classification scheme, it places a Staphylococcus within this order Bacillus. So Bacillus are the order is the order which is comprised of both rods and cocci. So basically, genus Bacillus represents the rod-shaped organisms, but not necessarily. The order Bacillus contains all the organisms with rod shape. So Staphylococcus is one of the important genus belonging to order Bacillus. So as you all know, and I hope that you have worked with Staphylococcus and you know that how it looks under microscope, and as if you have observed it, so definitely you should be well aware that these are basically grape-like clusters. These are cocci. They are, an arrangement is grape-like. And one of the important genus that the species that you have discussed is the Staphylococcus aureus, which is which is capable of generating a yellow color pigment, and in turn the colonies, they become yellow, so or golden color colonies. Basically its name indicates that its name is based on its ability of producing this yellow color pigment, because in the word Aries it represents a golden color. So they are mostly facultative anaerobes, and they can grow under high salt concentration, or must say high osmotic pressure and low moisture, so they can grow well. Other organisms, other bacterial species, they usually uh, grow either poorly or not, and they don't grow at the conditions of high osmotic pressure and low moisture, because this property is basically of more than the yeast cells. But in among bacteria, Staphylococcus, they show resistance in the high osmotic pressure and low moisture. Not only they survive, but they grow well under these conditions. So this is important regarding Staphylococcus. And the reason for the reason for, I must say, producing pigments, why any organism produce pigments. So definitely there must be a reason. Nothing is produced because of no reason, right? So the yellow color pigment is capable of protecting the colonies from the effect of sunlight. So antimicrobial effects of sunlight, because sunlight is, it contains ultraviolet rays, and which are antimicrobial in nature. So this pigment, it protects the colonies, right? And because of their capability of growing at high osmotic and pressure and low moisture, they are able to grow in your nasal secretions. So basically they harbor your, they are the common inhabitants of your nasal cavity, and the common inhabitants of your skin. So both the places, they offer high osmotic pressure and low moisture. So that's the reason that you can find these Staphylococcus strains at these places in your body. So as Staphylococcus aureus, it can be pathogenic if it targets the bones, right? So because it's capable of producing a toxin, and that toxin is very severe, and that toxin can cause a disease, or which is a problem which is known as toxic shock syndrome, right? So this is a very severe infection, and it may be fatal sometime, and it is characterized by, you can say, high fever and vomiting, and sometimes death occurs. And because this toxin is Staphylococcus aureus also produces some other toxins which targets your gut, right? So it is also the cause of your common cause of the food poisoning. Staphylococcus aureus based food poisoning is very common, because this organism, if it is growing in your food, it generates toxin. Cells are not the cells, viable cells, that do not display the pathogenicity because they are killed in your stomach, but the toxin is capable of causing damage to your gut, and it results in the food poisoning. So this was all about Bacillus, in which we discussed Bacillus, genus Bacillus, and the genus Staphylococcus. Then we will discuss Lactobacillus, which is also a very common and commercially important or industrially important order, Lactobacillus. So one of the prominent genus in this is Lactobacillus, which is commonly present in your body. And where it is located, it's located in the intestinal tract, oral cavity, right? And there are strains, Lactobacillus. There are different species of Lactobacilli which are used commercially for the production of fermented products like pickles, sauerkraut, buttermilk, and yogurt, right? So they are used as starter cultures, and they are used for fermentation of these fermentation processes, and the products are mentioned here. These are few products. There are a lot more products, right? Another thing, or another important genus is genus Streptococcus, which belongs to the order Lactobacillus. Although, as you can see that if you look under microscope, Staphylococcus, you might, in the morphology might confuse you, but their DNA analysis, it placed them into entirely different orders, right? So they are spherical and gram-positive bacteria, but they typically appear in chains. So their typical arrangement is in chains. But what happens when you take a lot of culture or inoculum is rich. So what happens that you or your smear is thick, so you find them in the clusters, the chains they cluster together, and you are not able to differentiate between Streptococcus and Staphylococcus. Right. Secondly, Streptococcus is capable of producing many enzymes, extracellular substances that can destroy the phagocytic cells. That ingest them. So I'm talking about pathogenic streptococci. Okay. And they can invade from your surfaces to the deeper connective tissues by basically dissolving the digesting the connective tissues of the host, and it results because of the different enzymes produced by Streptococcus species, pathogenic Streptococcus species, and they also result in extensive tissue destruction. So in general, Streptococcus can be found in your oral cavity and intestinal tract, but they may cause different infections because of their high potential of producing the enzymes capable of hydrolyzing the connective tissues. So they can be allowed to spread from the site of injury, and basically they can produce uh, enzymes that can lyse the fibrin, or which is thread-like protein, and they can easily they produce so some of those that enzyme that can dissolve the blood clot. Basically, blood clot is formed when fibrinogen is converted into fibrin, right? So basically, when fibrin threads they start accumulating at a place of injury, what happens? A clot is formed. Basically, it's part of healing process of your body. But the Streptococcus species, they are capable of producing the enzymes that lyse that fibrin. And when that lyse that fibrin, so definitely there is a chance of a spread from that area or from the injury area to the other sites as well. So there are certain streptococcal species that we'll discuss, and you can differentiate the streptococci on different species of streptococci whenever they are grown on the blood agar, because they show a pattern of hemolysis. So blood agar contains blood, and blood contains RBCs, and when RBCs uh, and blood is there in agar, the color becomes red. Red color because of the RBCs. So what happens if any organism is capable of lysing the RBCs? So what happens that from that site the color becomes colorless, right? So basically, whenever this lysis it happens, so there is a clear zone of hemolysis on the blood agar. So this hemolysis can be of two types. There may be a complete hemolysis or there may be partial hemolysis. So there are some species which can hemolyze the RBCs completely, and so which is called as beta hemolysis. And beta hemolytic species, or the species which are capable of beta hemolysis, they produce hemolysin, and because of that production of hemolysins, RBCs they are ruptured and they are killed, I must say. So there is a clear zone of hemolysis on blood agar around these colonies, as it's indicated in the picture. So these are the streptococcal species growing, and they are also known as the beta hemolytic streptococci because they are capable of completely killing the RBCs. For example, we have Streptococcus pyogenes. One of the examples that you have heard about is Streptococcus pyogenes, which is one of the beta hemolytic species. So on blood agar, if you have, for example, two strains, one of that is Streptococcus and there is another strain. So if the two they are mixed, somehow the culture is mixed, so you can separate them on simply cultivating or the culturing streaking them on the blood agar. So those colonies giving you the clear zone of hemolysis, they will be of Streptococcus pyogenes. So the Streptococcus pyogenes is very much important in that this organism causes severe infections. So this is also known as flesh-eating bacteria because definitely it causes massive destruction in your tissues. So, and this is very uh, important to discuss that how they escape your immune system, right? Because this Streptococcus pyogenes uh, has a protein on its surface which is known as M protein, which is one of the most important virulence factor of Bacillus of Streptococcus pyogenes because of this protein it can avoid phagocytosis, right? And once phagocytosis is avoided, so means that you are actually uh, losing one of the major defense action, and this is very powerful in that aspect that it can escape phagocytosis. So I told you that there are two types of hemolysis: complete hemolysis and partial hemolysis. So one of the type is the partial hemolysis, and this is also known as alpha hemolysis. So when the RBCs they are partially lysed, what happens that there is no clear zone of hemolysis. Rather, the partial hemolysis results in the green greening color around the colonies. So it indicates the partial hemolysis. So if you notice green color or the colonies around green color around colonies on the blood agar, it indicates the partial hemolysis or the alpha hemolysis. And one of the best known example is basically Streptococcus pneumoniae. So it is another very common strain causing pneumonia, and this partial destruction is because of the production of H2O2 by these strains, so which targets the RBCs and results in partial destruction. So if you get two cultures and they are mixed, so I was talking about Streptococcus pneumoniae and Streptococcus pyogenes because these are important pathogenic strains. So you can discuss or differentiate them easily on the blood agar. That's why blood agar is also known as a differential medium, because you can differentiate the two species on the same plate, right, by looking at the pattern of hemolysis. Right. Another important strain is Streptococcus mutans. And Streptococcus mutans is the primary cause of your dental caries, right? So this is all about the Streptococcus. We are discussing the order Lactobacillus, and in that we discuss first Lactobacillus, then we discuss Streptococcus, and now we are discussing the Enterococcus. So as the name indicates, these are the cocci, and they are restricted to the areas of the body which are rich in nutrients but low in oxygen. Definitely, the gut represents the best example of such region. And other than that, there is oral cavity and the vaginal cavity. So since they are present in the gut, so they are also present in the stool, human stool, and they are also on the cause of nosocomial infections right, or hospital acquired infections, and they also confer high resistance to the antibiotics. And one of the uh, two of the important species includes Enterococcus faecalis and Enterococcus faecium. These two species, they are pathogenic species, and they are responsible for mostly the infections of surgical wounds and the UTIs, urinary tract infections. So then we have Listeria. You have heard about Listeria, and this genus is very much important because this one of the species of the genus Listeria is the Listeria monocytogenes. It can contaminate your food, it can contaminate your dairy products, and interestingly, this organism is not capable of surviving only in the phagocytic cells, but it can also grow at the refrigeration temperatures. So since it can target your food, and it can grow at the refrigeration temperatures, which is one of the main reason for spoiling your fruit products when they are kept in the refrigeration temperatures. Secondly, when you consume that food contaminated with Listeria, so it can survive within phagocytic cells. So it is strong enough to cause serious problems, right? And it specifically targets if it targets the pregnant woman, so what happens that this organism, it poses a threat of stillbirth, and it causes serious damage to the fetus. So this is another well-known genus of the order Lactobacillus. Now, another important order is the Mycoplasma tails, and Mycoplasma tails, uh, they were previously considered as viruses, but now they are grouped in the gram-positive organisms uh, because of their 16S rDNA. But the remarkable feature is that these mycoplasmas, they lack cell wall, and they can produce the filaments that resemble fungi. And since they lack cell wall, they are very small, and their size is very small, that you cannot see its colonies by your naked eye. So generally, you can see bacterial colonies in the agar plates with your eyes, unaided eyes. But the colonies of mycoplasma, they are also visualized under microscope. So it's very small that its colonies, they are also microscopic colonies. Right now, one question is that if they lack cell wall, then how they are placed in the gram-positive organisms? Because gram-positive and gram-negatives, the decision that where to place any organism is based on the cell wall chemistry. That's if cell wall is showing gram-positive reaction or it's showing gram-negative reaction. So how this organism lacking cell wall can be placed in this group? So the answer is that this placement is, I told you multiple times, is not because of the phonetic characters, but because of the phylogenetic characters. Phylogenetic analysis placed them into this volume. So they're very small, and they range in size from 0.1 to 0.25 micrometer, and they're so small that they can pass through the filters that can retain the other bacteria. So they are very small self-replicating organisms which are capable of free living existence. So they're free living organisms. There are few pathogenic strains. One of that is Mycoplasma pneumoniae, which is a common cause of mild pneumonia. right? And now one of the questions should arise in your mind, that cell wall is very much important while for maintaining the osmotic pressure and protection. So how they are surviving without a cell wall in any environment? Because if any organism it loses cell wall, so it results in eventual killing of that cell because of the osmotic pressures of the environment or the environmental stresses, right? So basically the membranes, they have a different composition. So they have a rich content of sterol in their cell membrane, which provides the same resistance against the environmental stresses, right? So one other another genus of this order, Mycoplasma is the Spiroplasma, and Spiroplasma basically they show cork screw morphology, and they are the serious plant pathogens, right? So you need to uh, get some responses of plant infections caused by Spiroplasma on the WhatsApp group, right? These are the colonies, and their colonies are also very distinctive, right? So I told you that if you want to grow mycoplasma, then you have to provide sterols in the medium. If you want to grow them onto the artificial medium, so that medium must be supplemented with the sterols, because the sterols must be incorporated in the membrane, right? So and you can see that they show very distinct egg-like, fried egg-like colonies. So their colonial appearance is also very different and differential from the other bacterial colonies. And even colonies are less than 1 millimeter in diameter, right? So they have fried egg like appearance, right? Which is very unique in that sense, that you can easily differentiate and notice the mycoplasma under microscope, right? So this was all about volume 3, and I shall stop here, and I'll make another video lecture for you comprising of volume 4.